Started. Let's quickly go around the room and actually introduce ourselves, make sure everybody knows everybody, because we got a couple new faces. I'm Sue Brantley. I'm the director of the CZO.
I'm Lily, Civil and Environmental Engineering.
Margot Kaye, Ecosystem Science Management.
Dave Eisenstadt, also Ecosystem Science Management.
Jason Kaye, also in that department.
Elizabeth De La Reguera. I'm a prospective student of Jason.
I'm Melissa Miller. I'm a master's student in soil science in Henry Lynn's lab.
Ken Davis, meteorology and atmospheric science.
Virginia Marcon. I'm a PhD student in geoscience.
I'm Yuting Kim, a postdoc in geoscience.
I'm Warren Reed. I'm a PhD student in the ecology program.
I'm Yuting in Department of Meteorology and Atmospheric Science. I work with Ken Davis.
I'm Wei. I'm a PhD student. I'm working with Dr. Lily.
I'm Dacheng Xiao in Department of Civil and Environmental Engineering. I work with Dr. Lily.
I'm Ted Primka. I'm an ecology PhD student.
Hi, I'm Joan Marie Delvecchio, and I am a master's student in the geosciences.
I'm Beth Hoagland, and I'm a PhD student working with Tess Rousseau in geosciences.
Hi, I'm Jennifer Williams. I'm the program coordinator.
Hi, I'm Callum Wayman. I'm a PhD student in the geosciences working with Tess Rousseau.
Hi, I'm Josh Woda. I'm a master's student working in geosciences.
Hello, I'm Dan Chapich. I'm a data manager at AZ.
First, a couple of announcements. There's a new eco-hydrologist that's joining Penn State. His name is John Duncan. I think he's going to be here in the summer, or at least in the spring. He's listening, I believe. He wants to help us, and he's really interested in implementing the SCAN unit. So that may be a nice contribution. Some other announcements. We're going to be the site for the annual field conference for the Pennsylvania Geological Survey in the fall, which is actually a pretty big deal. There's like 250 people come. And it's going to be great. But what's hard is we have to come up with 10 people that can be there all day in the morning and 10 people all day in the afternoon, because they're going to be moving people around stations. And so we're really going to need some help in order to be able to man some stations. And it's going to be on October 6th. So we're going to send information out about that. We may have to get brothers, sisters, friends, anybody to man these stations, because obviously that's a lot of time. Okay, but I think it's a good thing. It's another one of those things that we can talk about as having impacted a lot of people in the state. Second thing, I would like to see if we can host this seminar. We only do this 3 or 4 times a semester, and we always do it at the same time. Whether we could do it on some Mondays from 12 to 1:30. Presumably we'll lose some people at that time also, but would that be another, like, could 2 of them be on Mondays from 12 to 1:30? Would that be a, I know we already talked about Ken likes to run. Is Ken's running the major problem? There's a weekly geochemistry seminar that I would like to be able to go see. So we thought we'd do half and half. So let's see if we can move to have some on a Monday, but we'll work it out. We'll let you know. Okay, that's great. Our all hands annual meeting is going to be all day the 11th and 12th. And we're actually, I think we're talking about moving some of the talks to the first day. I'm mixed up now because I'm not looking at the whole agenda, but we were going to move up some of the talks and spread them out so we have a little bit more time of discussion. Because usually we have a day that's almost all talk and then a little discussion at the end. I think we're going to try to move some of the talks a little bit earlier in the week. It's still during the all hands time so that we can have some discussion before noon, just to get us talking about renewal. We think that there's going to be a call for renewal in the summer or the early fall, and we want to start getting people's heads wrapped around ideas for the renewal. Okay, so we'll send out some proto-agendas and just wanted to get you to think about that. The other thing is we have this idea, we keep putting the data into the data site, Dan works really hard to wrangle it out of you, put it up there so that it's really easy to find. But then we have people come in like Ted, who tries to find data, and then we find that it's not so easy to find. Or you can find the data, but it's hard to make it work, or there's mistakes in it. And we've kind of realized that, you know, the way you find the mistakes in the data is you actually pull it out and you use it. And so we have this idea that for the all hands, the students can self-organize into making some posters that show the data. Are you willing to talk a little bit about what the idea is, Yuting, that you're going to try to organize? Can I give you the...
I think the idea is that we work together and we come up with some questions and things we want to understand about the data. And those questions, like what data can we use to answer those questions? And then we can have a draft of those questions and we send to the faculty and let them help us revise it. And so we can work in groups, like collaborate to explore the data and to visualize the data. And that can help us to, you know, find the mistakes, help us to quality control the data and meet our need. And maybe I think in the near future, after we did this, we can come up with some standard procedure to examine the data, maybe quarterly or monthly, to, you know, identify the mistakes in a timely manner, like visualize the data. I think that's helpful. I like your idea about comparing the SEPFlux data and the flux tower data. And I'm also interested in the soil respiration flux. So those carbon fluxes data, we can compare like their magnitude and their spatial temporal pattern, things like that. And Sue mentioned about the groundwater seasonality and also the SEPFlux. So we can come up with different questions from different like discipline. Yeah.
Sounds good, but I assume the questions are simple questions like, how did the data look this past month in this, not like your dissertation question, right? Okay. Yeah, all right. That sounds great. Well, if she can talk the whole rest of the group, and yeah, that'd be fine. But
But we also are going to really encourage the grad students plus postdocs to self-organize some posters that pull together big data sets and pull them out of our data system, which we are hoping will allow us to find some mistakes and things, but also to find some science, to answer some scientific questions. Does that kind of make some sense? Oh, yeah.
The other part of it is that as you're, say, trying to get the leaf litter data, where the heck is leaf litter data? I have to find it. Then you should let Dan know that and basically document, because we want to make the website accessible. We want to get stuff off box and into the website so that we are fulfilling the NSF aspects of our data accessibility. And we originally just thinking of having someone check out accessibility, but you can blame me. I felt that there was an opportunity here to actually start looking at our data a little bit and troubleshooting mistakes.
right. Hey, look what last year looked like. Let's look at these things and just, wow, that's cool. What's going on?
That's what I'm thinking. I mean, something that's only taken up one time, I think that question might be sufficient, but things that are taken across the season, we can just make a plot of the season. This is what the eddy flux did, this is what the soil moisture did, this is what the water flow did.
We're so good at this microphone thing. Yeah, exactly. And I talked to Virginia a little bit about pulling all the soil chemistry data so that we'd see the Shale Hills and Garner Run together, maybe with Al Den's cosmogenic isotope data, just kind of pulled together for the first time. And, you know, can we find it online and where are the mistakes? Some of this is because of you, Ted. You did such a wonderful job pulling data out, and then you found some mistakes, and it got us thinking that that's really the way to find problems in our data sets, is to actually pull out data that you're trying to use. So that's what we're hoping we can do. And maybe you would be on this soil chem, you know, I don't know, like you guys can talk about what these posters would. Okay? That's for all hands, which is May 11 and 12. The talks are going to be as they have been in the past. Each student or postdoc for a given hypothesis will give a talk about the cool stuff you've been doing and the new ideas you have. But we're also going to ask everybody to say, and this is what we could do in the next 5 years, kind of thing. In other words, to look about what you've done, but then at the end, think about what could we be doing into the future. Because we really want to use the all hands to be thinking about the future as much as thinking about what we've accomplished so far. Okay? We'll send more emails out about this, but just to get you to start thinking about it. And, you know, we were really honestly hoping that we'll really have some wild and crazy ideas, you know, like really get people thinking. We're kind of... Wild. I can't even remember how to do it. Wild and crazy guy. Probably nobody here even knows who that is, Dave. See what I mean? Okay. So those are all my announcements. Jean Marie wanted to talk too, because we have another ongoing thing that she's working on.
Sure. Yeah. So just briefly on. So if you've been keyed into the couple of emails that have been sent about our collaboration with Shavers Creek Environmental Center, basically we chatted last fall about how we could better communicate the science that we're doing, which is within sort of the environmental center's trail system, you know, what they consider to be part of their ecological educational system, whereas we have a much more like sort of hard science action going on. So their idea was, hey, you know, maybe we could start a dialogue, figure out ways to communicate, work together to communicate to visitors what science you are doing out there. And the conclusion of the fall semester's worth of discussions and site visit was that perhaps we assemble a list of places along the pre-existing trails at Shavers Creek that demonstrate the work done by CZO scientists. What's convenient is that along a couple of trails, which I've highlighted in yellow here, circles around a lot of things that are important to the CZO. It passes by some like sensors and other things where places where measurements are being made, in addition to interesting ecological, geomorphological, water cyclological places. So the outpouring of this for this semester is that the Shavers Creek Environmental Center hosts Penn State undergraduate students who are interested in being environmental educators, people working in recreation, education outreach, environmental education. And so there's a class that they offer where they instruct students to design and implement their own educational displays or interpretive material. And so we're working with the folks who are running this class to use the CZO sites that we had previously identified as the basis for what these students are doing in their class. So the first call for you all is that we are still seeking sites or place-based demonstrations of your science that demonstrate major CZO themes. We already have a couple up here on this map. For example, there's an old weir at the watershed above the CZO subcatchment. There's parts of the landscape as you walk around where you can see that there's been deforestation and regrowth. There's interesting geology, there's interesting water things, and so we've identified those places. But if you know of your group, or if you can kind of point on a map and say, it'd be really interesting to demonstrate the work that I do with this particular species of tree or this particular, you know, landscape feature, that would be really useful. And if you have those ideas, please still send them over to me or Jennifer, and we can let the Shavers Creek folks know that there are those sites. In addition, we're also, Oh, okay. So, and I mean, we had this discussion where we didn't exactly want people to be walking around the subcatchment where all of our sensitive instrumentation is. So we were sort of focusing on places just outside of our technical sort of study site so that people wouldn't be kicking over lysimeters. So that's the motivation for keeping it not within the subcatchment itself. Mm-hmm. Right. Yes. And one of the other aspect that we're seeking your all scientists help for is involvement communicating with the students how they should be conveying the science that you're doing. So these are undergraduate non-science majors, right? They're doing like recreation and sort of like environmental education. So they will need help. You can't just send them, you know, your most recent paper in the journals and they know what work you're doing, right? So we need people to help distill your science information to them so that they can then interpret them for not only themselves but for visitors. So the Shavers Creek staff have asked that, you know, if we could either, best thing would be to come in person, tentatively scheduled February 16th, though it depends on availability from our end of things. That's when Shavers Creek is good, but if you are all not available. So yes. Yes, so they would love it if we came out and could physically be there to answer the students' questions if we give them this list ahead of time. That deadline is tentatively like February 6th, so if you had more ideas, that would be February 6th. If you, oh, sorry. So to get us your final thoughts, we would have a final list of sites by February 6th. And then the sort of site visit for the scientists is planned for February 16th, tentatively. And so if you could make that, that would be awesome. Yes, Ken. What? Sorry. Yes. Right. Yeah, the real-time sensing thing, my understanding was that that was slightly separate from this, and like that was not necessarily this. Yes, the students for this semester were instructed to do like this part of that project. I think the sensor side of things might be for a later date. No? Okay. Mm-hmm. Okay. Mm-hmm. Mm-hmm. Right. Right. We've highlighted a couple of you know, overarching themes or ideas that have arisen, such as the idea of the carbon cycle, the idea of the water cycle, the idea that geology shapes the landscape, and the idea of human-environment interaction, humans impacting the landscape. So those were just some of the example big ideas that came from our site discussions. So in terms of still helping the undergraduate students who would be designing these displays, helping to answer questions in person on the site visit day would be great. If you can't do that, at least saying, hey, I'm the person who knows what these sensors do, this is my email, so that the students can say, I have no idea what a mini risotron does, who can I ask to find out? And then they have you as the contact. And finally, if you have sort of non-technical resources that, you know, printed, you know, if you have websites or, you know, PDFs explaining in a non-technical fashion what it is that you're doing, that would be another like great resource that you could send to me, and I can send it off to the students, because I can easily send things on sort of geomorphology, but I don't sometimes know what other people are doing in a non-technical sense. So these are things that will help with their class. Okay.
put together in the wherever it is or
I would suggest that you write the idea that you think is interesting, teach people about it. And your students are supposed to learn what they're about communicating. You say, this is a cool idea, we should teach people about it. And then they say, we like it, come and talk to us about it. And they can figure out what it is. Probably not. If you just said water cycle, they might say,
Water psychology, yeah.
Yeah, I mean, maybe there's something you learn with modeling that is actually a concept that others would be really, that's the kind of it's really extreme that.
Well, I think one thing is that we're talking about the ideas that are happening. This is necessarily like a place like modeling, especially is a good example. You just want to talk about these ideas. Yeah, so I mean, if we want to talk about these ideas that aren't necessarily site specific or anything, I mean, that's a good opportunity to add more things to a potential exhibit, like within the actual environmental center, and have, posters or, something like that. Maybe show a short video of, I don't know, a model running or something like that. I don't know, like something like that. Right. Yeah, exactly. Stuff like that. So stuff that's not site specific, but that you can put inside in the center and can be explored by students and stuff. Because I mean, obviously the center is going to be the home base for all these lessons, I assume. So, yeah.
Other questions? I think that's all our, those are just our announcements. Okay, so now we're going to do something about nitrogen. I'm not sure who's in charge and what the order is, but you can take it away.
I don't think any.
On the list of whatever Jennifer.
Oh, okay. It was Yuting
Well,
and
what
Yuting.
makes the most logical sense? I think Yuting and Yuting will be both talking about English. I'll be presenting me too.
My models are conceptual, so I'm different than the other one.
So it's unique when you talk to me.
He sent me the 1.12.
Maybe you can take charge. You never go first, but take charge. Tell someone to go first. if you want to do it.
Take 1/2 of the hour. No, 20 minutes. You should take 20 minutes for more time.
Okay. Yeah, the first one. So this is the outline I have for my presentation. I'll first talk about the nitrogen modeling in PBGC, and then I'll talk about Yuting's work on coupling of the PBGC with Flux Team. And then there's a few slides on some issues with nitrogen modeling based on some paper. Oh, thank you. Okay. And first, I'll talk about the nitrogen modeling in PBGC. So in PBGC, a very crucial nitrogen pool is soil mineral nitrogen. And the nitrogen deposition and nitrogen fixation are inputs to the model. And they are fixed rate provided by the users. If you have the observation, you can just directly tell the PBGC what number it is. And then nitrogen can get lost through the denitrification and nitrogen leaching. And both of these two are they are lost at a constant rate, a constant fraction of the nitrogen pool. And then some mineral nitrogen can be uptaken by plants, and it can also be immobilized by microbes as they decompose the litter and soil organic matter, and can also get supplied through the okay, can also get supplied through this mineralization process. And sorry. So this part is the most complicated part in the model because it involves the carbon-nitrogen interaction. And then I will talk about the denitrification, nitrogen leaching at the end. So this is the carbon cycle in PBGC. Atmospheric CO2 comes into the ecosystem through photosynthesis, and some of the carbon got lost through maintenance respiration and growth respiration. And then the rest of the carbon get into different plant compartments like stems, leaves, and roots. And then as the tree die or litter fall, then become litter, and microbes decompose them, become soil organic matter, and further decomposed by the microbes, and CO2 will return back to the atmosphere. Okay, so maintenance respiration, growth respiration, growth. GROWTH. Sorry. Growth. Huh? Yeah. And heterotrophic respiration. Yeah. And then this part is the nitrogen cycle. See, they couple together at this part. So atmospheric nitrogen get into the soil minor nitrogen through deposition and plant root fixation. And it would uptake by the plants and enter into different compartments, and then later fall, enter into later carbon pool, and then decompose into the soil organic matter carbon pool. And as they decompose, as the microbes decompose the litter and soil carbon, it can deplete soil minor nitrogen pool through immobilization, because there might be a demand for nitrogen to decompose the litter, or it can replenish soil minor nitrogen pool through mineralization. And as they mineralize as the microbes mineralize the soil carbon litter, it can get nitrogen loss through the gaseous emission, denitrification. And this is a similar diagram showing you that the soil minor nitrogen pool can be uptaken by plants and can be taken by the microbes to decompose the litter and soil carbon. So trees and microbes are competing for nitrogen in this model. So these two are the main competitor for nitrogen. So yeah, happens afterwards. Yes. So I'll show you how PBGC is calculating the nitrogen immobilization or mineralization. This diagram shows you the different litter carbon pools and soil carbon pools, and they cascade down by the decomposition rate. So as you go down, it's getting harder and harder to decompose because the litter quality gets lower. And so, for example, we have litter carbon pool 1 that have a higher quality. Yeah, like the fresh litter. Yeah, fresh leaf litter. We call it a donor. And then as it being decomposed, a fraction of the carbon get respired and lost back to the atmosphere. And then the rest of them enter into the soil carbon pool 1. We call it acceptor. And then we calculate the nitrogen immobilization based on this equation. So basically, I forgot to mention to you about this. Each pool has a fixed CN ratio. So this one suppose has a CN ratio of maybe 18. I just give you a hypothesis number. And this might have a CN ratio of 12. Yeah. Okay. Logic for yeah, I think so. Wait. Decomposition time. Yeah. So different pool. They're conceptual. They're conceptual. Yes. So for different each leader pool, they have a different fraction of lignin, cellulose, and what is the other? Yeah, and labile. Yeah. So they have a fraction of them. And also the more pool, the more promise. I don't think CN ratio in this model linked to the decay rates. No. Of each different carbon pool. Yeah. And for each different plant compartments, they also have a CN ratio. Yeah. Like leaves, they have a CN ratio, a fixed CN ratio in the model. And then liter has a fixed CN ratio. And woody, yeah, have a fixed CN ratio. They all, and roots. Yeah, this is if the demand of the acceptor for nitrogen is higher than the the supply, then there will be immobilization happening through this process, decomposition. So it will get more nitrogen from this soil minor nitrogen pool. The soil minor nitrogen pool is like a buffer. When there's need, then it's supplied to the microbes. When there's like excess of nitrogen, then it will go back to replenish the soil minor nitrogen pool. And yeah, so this is when there's the soil minor nitrogen pool is greater than plant demand, so the model will also calculate. So this part is from the carbon cycle. When there's a plant demand and the soil minor nitrogen pool can meet that demand, then it can fix all the carbon that can. So it means it's not limited by nitrogen. The photosynthesis is not limited by nitrogen. But if there is a larger plant demand, then nitrogen here will limit the carbon fixation. And it not only affects the tree growth, the plant, it also affects the decomposition. And I plot 3-year nitrogen demand of plants and microbes. So you can see the microbes always have a higher nitrogen demand than the plants. Model, yeah, model 3-year. Yeah, I have the seasonality. So in winter, there's nothing going on there. And here, the model calculate the plant demand and also calculate the microbe demand. And then it has a like it has a number of like how much nitrogen is available there, and we'll split this pool into to meet different needs according to their need. Like if there's more need for microbes, then they will give more nitrogen to microbes to decomposition. Yeah. Huh? Adequate or any? Oh, I mean they split, like there's a ratio of this too. So yeah, so if there's more demand here, then we'll give more nitrogen to microbes, but their plants still get some according to the ratio of the demand. Yeah. And here's a plot. It's also okay. This is model, and the black dots are observation from the tower.
So this is the model for Shield Hill?
Yeah. So yeah, and measurements. And for 2009, 2010, and because the measurements only start from May 2009, and the data, like we lost data after 2010 October, I think. So this is the summer, winter, summer. And the y-axis is the NEP. So positive means there's CO2 coming into the ecosystem. And this is respiration. And net ecosystem productivity. So how productive the ecosystem is. And so here we use the default nitrogen deposition, and here we use an observed nitrogen deposition rate. And the magnitude of the NEP in the summer increased and almost can match the flat tower observations. But the problem is in winter time, there's also more decomposition going on because the microbes, they have enough nitrogen. And this is contrary to some observations that like from literatures, and they suggest when there's plenty nitrogen, the heterotrophic respiration is tend to be inhibited. The model doesn't consider, right? Yeah.
What are those observations from?
Tower. Yeah. These are the flux tower from the flux tower, eddy covariance.
And you're measuring the carbon flux in and out of the watershed?
Yeah.
You gotta really break this down for me because I didn't know the tower was measuring anything. Okay, so it's measuring carbon going in or out of the.
Yeah.
When it's positive, the carbon is net leaving the catchment, and when it's negative, it's net.
No, this is summer, so I think the carbon is going into the ecosystem.
Okay.
They are making photosynthesis.
Negative, it's net going out.
Yeah, respiration.
So are we saying that the black dots in the winter are wrong?
Yeah, I also agree there's some problem in the observations because so the flux tower, one hypothesis to make the flux measurements is that there's no net sink or source of the air. So over a period of time, average, no lateral, but there's on the mountain and in the winter time and also in the nighttime, the boundary layer is pretty shallow and very stable, and the air is now well mixed. So the sensor will sense a net flow, like when the cold air going down the valley, the sensor will think, oh, there's CO2 going into the ecosystem, right? Like, I mean, the CO2. Mm. Yeah. Mm-hmm. Oh. Yeah. Oh. Okay. Yeah.
You can't correct it. But it would seem to me that you need sensors along the lateral flow path. That's what. I'm totally surprised. I think it's
The question, so during the growing season, you're not having the same underestimation of respiration. So you don't have to move all the blocks out, or do we? I mean, because is there more mixing? You know, I mean, so if we're saying we've got this issue of boundary layer and what is leaving the ecosystem is never reaching the house. That's a 10. In the growing season, do you have the same problem? That you have respiration coming out that never makes it in the tower.
I think nighttime will be an example. Right.
So then those need to be corrected as well.
But you've done something about that. Right? You have a correction.
Yeah, I did that. Or fill the gap. Yeah.
Is that what you're saying? Okay.
He doesn't like the topography. It's the topography that is the problem.
Right. Okay.
But I mean, as a cynic, the previous slide showed 9 boxes with different parameters, presumably. Like, did we fit this? Like, did we just turn knobs until we got the good fit? That's what I didn't understand. We went from this box model to this simulation. Well, you had your boxes. So you didn't change any of the parameters. Okay, so you didn't change any of the parameters in the model, you just changed nitrogen deposition.
Okay. Move on. It's not working. Oh, wait. Oh, oh. Thanks. Yeah, now it comes to the nitrogen leaching and denitrification. And both processes are assumed to occur at a constant rate. So denitrification will assume 1% of night mineralization in this process, 1% is lost as gas. And then if there's some days that we have excess soil manure nitrogen that exceed plant and microbe demand, then 50% of the nitrogen in that day will be lost. Oh, sorry. And then nitrogen leaching is like 10% of the nitrogen concentration times the water outflow will be leached out of the ecosystem. And that's the like how PBGC simulate nitrogen cycle. And then I'm going to show you Yuting's work on coupling PBGC with Flux Team. So Flux Team is a 3D simulation of the hydrological cycle. Give us soil moisture, soil temperature, and PBGC is just one point. It doesn't simulate lateral communication with the grades, different grades. Yeah, so if we couple these two models, we can have a three-dimensional carbon, water, nitrogen cycle in the model. So this is Flux Team Yuting coupled for his PhD dissertation, and it can simulate the vertical and lateral groundwater surface and subsurface water flow, and also the energy cycle, energy budget here in the watershed. And PBGC, I just mentioned, has the blue lines are the water cycle, and the red lines are the nitrogen cycle, and the brown lines are the carbon cycle. And in this coupling, he removed all the blue lines coupled with the Flux Team. And this part is Flux Team. So Flux Team gives PBGC the root zone soil moisture to calculate the stomatoconductance for photosynthesis, and then also provide the total soil moisture storage to calculate the nitrogen concentration for the leaching, and soil temperature, solar radiation. This is according to the topography, aspect, and slope that have a different solar radiation at different point. And then albedos, in PBGC it's a constant, but in this coupled model, it will change with the season. And then aerodynamic resistance, also it was fixed in PBGC, now it's changing with the meteorological condition. And then PBGC, it's wind. No? Yeah. I thought it's the boundary layer around the leaves, right? Well, this is from Flux Team, I think, this part now. Yeah. Previously, PBGC is just a fixed number per parameter. Yeah. And PBGC feed the Flux Team with LAI, the Ferry Index. And also, Yuting add this work to give the nitrogen transport along with the water flow. Yeah.
And so each of these are discretized or spatial.
Yeah.
So Tim decides whether the water moves from here to here,
Mm-hmm.
and then PBGC responds.
Mm-hmm. It is running at different, triangle. Yeah. Yeah. And even though you can simulate PBGC in the watershed, but if you don't couple with Flux Team, it's just they don't talk to each other. The grids, they don't talk to each other. Now they can talk to each other through the lateral transport. And these are the simulation at Shield Hills. Yuting used default BGC parameters without tuning any parameter, and only used deciduous broadleaf trees forest in this watershed, and running for 6,000 year uncoupled spin up by repeating this 3 years forcing. So for Flux Team, for PBGC to reach equilibrium state, it takes thousands of years. But for Flux Team to run 6,000 years, that will take I don't know how long. So, so this spin up is first spin up the flux beam and get the soil moisture, soil temperature ready to feed into PBGC, and then yeah.
I'm guessing that most of the room doesn't know what you mean by reaching equilibrium. You might ask, I don't know. Maybe everybody does know what you're talking about, but I'm not sure.
Okay, so for ecosystem model, it's necessary to run the model into equilibrium state. Means, okay, for PBGC, we assume just a little leaf carbon in the beginning of the time. And then from this little leaf, it starts to grow into a forest that has the criteria for PBGC is that the soil carbon pool remains steady state. So when you repeat this 3-year forcing, you get the same soil carbon pool at that time. So we call it equilibrium state. Okay. And then after we run PBGC for unto the equilibrium state, now we can run the coupled run for Flux Team BGC for these 3 years. And these are the inputs from the Flux Team to PBGC. Available root zone soil moisture. So you can see there's higher soil moisture in the stream and the swale. And also there's slightly wetter in the north facing slope due to a less evapotranspiration. And then the total soil moisture storage have a similar pattern, wetter in the swale and valley floor. And then solar radiation, you can see the big difference between south facing and north facing. And then soil temperature has a similar pattern with solar radiation.
Yeah, good. Okay, you may need to answer this. I asked the question actually when I looked at the paper. This doesn't seem to make sense to me if it's over the whole year. Was this just done over the growing season, these moisture patterns, or was it done over the whole year?
This is the average of the 3-year simulation. Can you hear me? Okay, so this is average over 3-year simulation, not just growing season, the whole year.
Yeah, so the plants will be unresponsive to soil moisture in winter. They're just not going to care.
That's right.
And similarly, in the question of aspect, are we thinking about it just in terms of its effect on soil temperature, or are we thinking about it in terms of PAR?
Aspect has its impact on both. So the solar radiation, actually, that's almost a fixed ratio with PAR. So solar radiation will reflect that.
But again, so the solar radiation at the lower angle in winter is going to be much less important than at the higher angle in summer.
Right. That's right. That's right. Yep.
Oh, go on. Oh, sorry. Okay. So this model simulation of vegetation carbon, soil carbon, leader carbon, and soil manure nitrogen. And you can see the similar patterns for all those pools, higher carbon and nitrogen in the soil and valley floor. Excuse me? Yeah. Yeah. He didn't do the parameter, as I mentioned.
We just can't go the forest like the mountains.
And there's more carbon nitrogen on the south-facing slope. And there's comparisons with observations, vegetation carbon compared to the above ground carbon. So higher vegetation carbon along the swale and valley floor, and also similar hot spots in the soil carbon. So the spatial pattern, we can get a little of the spatial pattern, right? But the parameters need to be tuned for get the magnitude. And there is also less spatial variation compared to observation. And this answers the question, what is driving vegetation carbon distribution in the watershed? So you need to plot the soil moisture, soil temperature, solar radiation, and soil manure nitrogen pool versus vegetation carbon. So you can see the soil manure nitrogen pool is the most important factor to control the, oh, sorry, I need to explain this. So the color indicates the elevation. So different color means different spots. So blue means in the valley floor, and yellow means on the ridge top. So you can see he plot other grades in this spot in these plots, and you can see soil manure nitrogen pool is the most important factor to control the spatial structure of vegetation carbon. So he plot this factor 1.95. I don't know what that means, but it's the most important factor to control the spatial structure of the vegetation carbon. And then the next one is solar radiation and the soil moisture. And the soil temperature is the least important. Yeah, the model. Yes. Yeah.
So the distribution of vegetation is controlled by soil mineral nitrogen.
Yes.
More than it's controlled by where the water is.
Yes. In this model. Yes. So nitrogen is coupled with water cycle. Right. And soil moisture is very important. Yeah. I forgot to mention one point. There are a few points here in the valley floor that doesn't fit into this line. And he suggests that these points in the valley floor are limited by light, not instead of by nitrogen.
Does that, how much difference does that make? Did you ever look at that? Because Ken just mentioned the lateral transfer is really important.
So in my study, I took 6 points instead of the whole watershed, and there's no lateral flow, but still soil manure nitrogen controls the vegetation carbon pool. But it doesn't mean that. Yeah, I also took the water, soil moisture from the observations. Yeah, yeah. But yeah, soil manure nitrogen pool directly controls the vegetation carbon pool, but water influence that nitrogen pool. Yeah. Yeah, you can tell off that lateral flow. Yeah. Yeah. Okay. And the conclusions we have already covered. And there, I don't need to cover this unless you want to discuss. There's some discussions. I can wait here.
Yeah, I can go
Mine, the second one. Okay, so Yuting and Yuting just talk about the essentially the terrestrial ecosystem and then soil biogeochemistry process. So what we are doing, this is work in progress. We have started this about maybe 2 months ago, looking at carbon, trying to add capabilities that we had before. We had RT models that have all the abiotic reaction processes like amino dissolution precipitation, ion exchange, these reactions. So we are adding in RT models soil biogeochemical reaction processes with emphasis on the interaction between the water and the different chemical species and organic carbon, nitrogen, and all that. So essentially, we kind of look more into the chemistry in the water and how these affect, for example, the reaction products and how much goes into different type of chemical species going into the water and become exported out of the watershed. So by doing that, I'm going to have a few slides, essentially 3 or 4 slides, outline the reactions that we think we have in mind for carbon, nitrogen, and phosphorus. So it's not just nitrogen. And then I will be talking a little bit about the reaction rate law we use and how it's coupled in terms of unsaturated zone and saturated zone in the RT model. So the soil carbon process, what we have in mind will be taking the calculation, like output of either PBGC or cycles. These have already calculated the total soil carbon, soil nitrogen, and soil potentially phosphate process. Probably not now, but we will be looking at how much soil organic carbon there. And once organic carbon is in soil, it goes through 2 major processes. One is the decomposition, and the other is the stabilization. So this is the color code here. When it's microimmediate processes, it's red. When it's abiotic, it's black. So essentially have 2 loops, major loops. And the decomposition process, it could be going through like a series of, for example, oxidation process, including those mediated by oxygen and other like nitrate, other electron acceptor related processes. And this decomposition leads to production of dissolved inorganic carbon or dissolved organic carbon. And the dissolved organic carbon can further become oxidized, become dissolved organic carbon. So this is the color coding in terms of chemical species, solid phase species, water, things that can go to water, and also the chemical species that go to gas phase, right? So dissolved inorganic carbon is essentially dissolved form of CO2 gas. And if it's accumulated to a certain extent, it can go to the CO2 phase and go to atmosphere. And on the other route, if soil organic carbon become absorbed on the solid phase, like through absorption on clay, they become more, we call recalcitrant organic carbon. So they become more stable and they accumulate. These become the organic carbons that will take much longer time to degrade or have long residence time. So we don't do the detailed pool, like 3, 4 pools. Is that your question? No. What's your question? We do. Alkalinity, that's what Sue's group, that's a geochemistry, aqueous geochemistry measurement. We have alkalinity, which is dissolved in organic compound. It will be very similar. Under certain pH condition, it will be very similar to alkalinity. DOC, no, not DOC. So DOC a different measurement, yeah.
The DOC is at the weir level or also at the soil sample?
In solution, we have the soil solutions as well.
Yeah, so right. So these data will be our constraint for the model eventually. And then for the soil nitrogen processes, what we have in mind is organic nitrogen. And we look into the detailed chemistry of the system. So mineralization leads to ammonium and nitrification to become nitrite and nitrate. And so it's going through this series of micromediate reaction. And there's also uptake of nitrite. These are blue species, these are water phase species. They can also uptake by plants, we call it immobilization, to become organic carbon again. So that's a sink for the aqueous species. And then nitrate can also go through denitrification process to become these gas phase species. Ammonium will be going through sorption, desorption processes that interacting with soil clay. So these are the abiotic reactions that we already have the capability. So the capability really will be adding these microbial processes in the RT model. And then for phosphorus, the organic, so there's the leaves and litter, all these have organic phosphorus. And I think Jason just told me an hour ago that we need dissolved organic phosphorus in the aqueous species. So it will go through the process of just going into dissolving organic phosphorus, but also in the water phase, there's also inorganic phosphorus that either coming from mineralization of organic phosphorus and dissolution of phosphor-containing minerals like apatite. And so these are the source of inorganic carbon. And the inorganic carbon could also resolve on soil clay to interact with these the soil minerals. So that's the reaction network we have in mind. And these abiotic process that I mentioned in these black arrows, they are already in RT, and we're adding these biogeochemical microbial reaction. And in general, they follow a rate law like this. The rate will depend on intrinsic rate constant of the bacteria, and then how much biomass is there, how much biomass mean microbial related bacteria, like bacteria related biomass, not like plants, roots, but it could be also, we've been learning a lot. This is, the rate could also depend on how much roots you have. So this is something additional we need to think about. And the rate will depend on how much organic carbon you have, which organic carbon providing the carbon source, and the electron acceptor essentially providing like oxygen, nitrate, or these different ion 3 mineral would providing the electron acceptor for the system. So these are the rate dependence. And then we are planning on having the whole series of biogeochemical redox ladder. So bacteria can use oxygen to get more energy, then they can use nitrate to get energy. So there's a whole ladder of biogeochemical reactions that we're hoping to put in. So if the system have oxygen, for example, and then not much of nitrogen, when oxygen becomes depleted, it will use other electron acceptors as for the biodegradation process. And it will also depends on temperature and how much soil moisture is in the system. So this will be the general reaction rate law we'll be using. for microbe-mediated reactions. And then this is my last slide. Essentially, all these reactions will be kind of folding the framework of PIM with unsaturated zone and saturated zone. So unsaturated zone is, so the PIM calculate in each prismatic grid, it calculates where the water table is. So the water table is the boundary between the unsaturated zone and saturated zone. So the unsaturated zone have essentially vertical flow from infiltration, recharge to saturated zone. And then the saturated zone is essentially the purged water table that have all these ladder transport processes. So Yuting talk about the nitrogen transport is a total nitrogen transport. We will be looking at the specific species like ammonium nitrate and the different species to looking at more details of aqueous geochemistry. And they're all folding the framework of PIM. So PIM will be still the driving force, and then we're looking at how the different chemical species interact in water phase and export of chemical species. That's all I have. Yeah.
So when you're talking about the latter biogeochemical reactions, you have oxygen and then sulfur down to iron and whatever. Do you only use oxygen first and then you move down to iron and then sulfur, or are all three reactions occurring at once?
It will be occurring all at the same time, but this term I didn't explain is we call inhibition term. So if oxygen is there, other electron acceptors are are not active in the sense, because this term will be small, meaning the oxygen will be inhibiting the use of other electron acceptors. So the reason we do that is bacteria can use oxygen to get more energy out of the system for their growth and the maintenance of life. And then once oxygen is depleted, this term will become close to 1. So then the ones that the next ladder become more dominant. So it's almost like a natural selection process. We would have all these processes in the system, and depends on which chemical is dominating the overall reaction process. Yeah, the overall rate. Yeah. Okay.
Yeah. So I'm going to be presenting observations. And I guess the way I want to think about this is whether these observations are useful for the modelers and benchmarking the modeling that you've done. And also, there's some questions that are hypotheses that could perhaps be tested by the model. All of what I'm going to present is Julie Weitzman's work, so we'll be publishing it soon. Julie started by thinking about whether we can use the sampling scheme that we've come up with in Shale Hills as a way to better understand nitrous oxide fluxes from soils. So spatial and temporal variability of nitrous oxide fluxes from soils is one of the wickedest problems in biogeochemistry of the nitrogen cycle. And so we wondered whether these katinas that we've been working on, and this would be like a swale katina in Shale Hills, might provide better prediction of spatial and temporal variability in nitrous oxide, where in the well-drained surface soils, where the light color is high oxygen soil and the dark color is low oxygen soil at the ridgetop, we would have very low nitrous oxide fluxes. Lily just showed that nitrous oxide is generated when in reduced conditions when nitrate is used as an electron acceptor, right? So if there's a lot of oxygen around, low N2O. Conversely, when you're really in the poorly drained part of the valley floor, the N2O itself can become an electron acceptor. I noticed that actually isn't in RT. I wonder if we should have nitrous oxide as an electron acceptor, because it gets consumed, unless it can diffuse away. Right. And in this case, so that was in that case, the gas leaves as N2 instead of N2O in these really reduced conditions. But in the sweet spot in the swale of the mid-slope swale, there's really flashy changing conditions where you have hot spots and hot moments. In other words, nitrate can be there, but then it becomes reduced because water's flowing into the system and nitrous oxide can go off. So this was a hypothesis. I'm going to tell you now it didn't work. Okay, so this did not afford us any new prediction of nitrous oxide, but we collected nitrous oxide data for 2 years on the planar slope and the swale frequently in the summer, and so we developed a pretty good budget for nitrous oxide loss, which is the next slide. Can you? I'm not able to advance. I am not able to advance. Okay, so and it's the table that I wanted to show. Sorry. All right. So with that data set, we calculated the total nitrous oxide flux from the soil, and we started thinking about how that might relate to other temperate deciduous forests that also have a good record of nitrous oxide flux. And so here we are at Shale Hills. The total amount we calculated to be lost was 0.7 kilograms per hectare per year. And we compared that to the Harvard Forest and to Hubbard Brook and to the Furno Forest in West Virginia. And we only have 4 sites, but we're speculating that maybe the medium clay content soils of Hubbard Brook and Shale Hills are a better predictor variable of the higher nitrous oxide fluxes than, say, precipitation alone or nitrogen inputs alone. Nitrogen inputs also vary, and the ages of the forest vary. But if you were to fit a line through these 2 things, you would get a really high R squared. Now, fitting a line through these 4 sites, and they're, you know, it presents a hypothesis that maybe the surface soil texture is determining the nitrous oxide fluxes from these eastern deciduous forests and not some of the other predictor variables that are possible. Okay, since we got to the nitrogen budgeting, there's a question? Yeah. Yeah, there is, yeah. Yeah, there's, I'll tell you what it is. So it is that, so if you have very high clay, which would be 35 or 40% clay, that's really the high end of the spectrum for clay. The soils become massive, and saturated soils only have anoxic environments. And so in this figure, that would be pushing it all to N2, right? It would be totally reduced. Not because of the watershed, because of all the clay, right? And so in intermediate clay, 20, 15% clay, you have a lot of microsite heterogeneity in soils. So you can have anaerobic sites and aerobic sites near each other. And that is a better condition for N2O flux. Yeah. I didn't understand that. So the soil physical properties.
Previous work that Yuting has led have suggested that if you get the soil physical properties wrong, your soil moisture simulation is terrible. And that's probably the most important thing for describing the spatial structure in soil moisture too.
Yeah, yeah, I think that's an interesting connection. Yeah. Okay, so since we moved on to thinking about the nitrous oxide as a component of the nitrogen budget, we built a nitrogen budget for Shale Hills with help from quite a few people. The term that we had measured in the last slide goes into, it's a component of this box, which is the gaseous outputs. Do I have a pointer? But we had to add N2 in, which we did really in a fudge factor way, let me just say. But anyway, so the total output, since it's mostly N2, that's impossible to measure, and some of it's N2O, the total output is about 3.5 kilograms per hectare per year. We also measured ammonia, which is negligible, and we think NOx is negligible. So that's an output. And the other output is stream flow, which is significantly smaller, 0.18 kilograms per hectare per year. The inputs are atmospheric deposition, which is measured at the NADP sites. Let me first see if I can Smart. Yeah. All right. The NADP site. So 7.1 kilograms per hectare per year. And another input that Sue and Liz Hasselmuller and the weathering calculations that have been done on the site enabled are weathering of the shale is an input of about 1 kilogram per hectare per year. We think inputs through nitrogen fixation are 0. And so if you take inputs and outputs, then the outputs are about 50% of the inputs. So this ecosystem is retaining about half of the nitrogen that comes into it each year. I think that's fairly typical of eastern deciduous forests, but one question is, where is that nitrogen going? It's either going to be the soils or the plants or some gas flux that we measured poorly. Really, those are the options, because I don't think we measured the water that poorly. And when we use the data from Lauren Smith's thesis and calculate nitrogen uptake into trees rather than carbon uptake, it's a really large number, 18 kilograms per hectare per year. It could more than account for this discrepancy of outputs minus inputs. So maybe trees are an important sink for nitrogen, and that would be a valuable thing to know in terms of predicting how this watershed retains or sends nitrogen downstream to the Chesapeake Bay. Yeah. We took tissue specific. How did we get the tree uptake of nitrogen? We found by allometric equations that were tissue specific. So leaves, twigs, stems, and we got nitrogen concentrations in each of those tissues. So it was allometry combined with tissue specific nitrogen concentrations. This is biomass at time 2 minus biomass at time 1.
A lot of nitrogen moving into I was just wondering about internal.
We didn't look much at it. We didn't look at all at internal.
So that might be
I guess, I'm not sure how it could be that much, but we should talk about that.
Okay.
Maybe we can refine that calculation. I am saying the trees are growing.
How about nitrogen loss as particles?
How about nitrogen lost as particles? You mean like parts of leaves flowing on in the stream? Sure, we haven't quantified that. That would be nice to add. It'd make the balance a little better. Yeah.
So can I ask, the carbon accumulation was not that you used to get the nitrogen accumulation?
No, it's not. I mean, we use the same DBHs, but
right.
We use different allometric equations.
No, that's okay. But you're using change in DBH to estimate.
Change in DBH for both.
But then there's decomposition.
The C-N ratio comes out to be about 200. Like if you take NPP.
I'm not questioning what what DBH data you did. But there's going to be decomposition of that carbon too. And I don't think that Lauren measured that.
We did need to think about mortality, and we looked at the number of trees that died. That's important. But you don't need to look at decomposition for trees growing. If we want to know.
What about trees that die or soil carbon?
Trees that die are important.
Okay.
Yeah.
Soil carbon decomposition?
Soil nitrogen and carbon decomposition are important. They're just impossible to measure over a period of 5 years.
Okay.
Yeah. How confident? Yeah. So the question is, how confident are we in the stream stream nitrogen exports? I feel like we should have a lot more stream nitrogen data. Julie pulled out these data from 2008 to 2010, where the dots are the stream nitrate concentrations, but we don't have a lot of DON, or we have very little DON or ammonium, and no particle nitrogen exports. So there are certainly gaps in that data set as well. So it's based on our nitrogen values are based on these nitrate concentrations.
Right. So I guess my question is that I've seen papers talking about a lot of these export of these depends on these large precipitation events. It will be flushing out a lot of things. So if you're missing these big events, it might be in terms of mass balance calculation might be.
We might be. I think this is ISCO sampled nitrate. I don't think we missed big events in this particular
Okay.
data set.
Okay. So we have it. Okay.
Okay. So that's interesting. There's a gap in the nitrogen budget that we could work to fill, and maybe models will help us understand it. Since we had pretty high nitrous oxide fluxes, we thought maybe that should arise from nitrate in the soil. So we went out throughout the watershed to these points, which I thought might be interesting, like Yuting. This is where we measured lots of nitrogen cycling processes, and we found a significant amount of nitrogen mineralization, but almost like near zero nitrification, net nitrification. So whatever's being produced by microorganisms is also being consumed by microorganisms in this measurement. We also measured the size of the nitrifier population, really, really low, and we measured denitrifier enzyme activity, which is like an assay of denitrification, very, very low. So we can't connect our high denitrification, our high nitrous oxide rates with high nitrate and nitrate production in the field. So that's another mystery I'd like to solve. Okay, one other way to look at the nitrogen data we have is in concentrations instead of mass balance, because I wanted to see whether atmospheric nitrogen had a high concentration and then that became low by the time it got to the stream, or the other way around. So here are the data plotted in a semi-confusing way because some of them go up and some of them go down. But the original plot was time and these dots, which are nitrate in the stream, and discharge is these solid lines. And so the nitrate in the stream follows this seasonal cycle that really reflects nitrogen limitation by the plants, with very low nitrate in the stream in the summer months, and then rising in the fall. And then the atmosphere, the concentration of nitrate in rainwater is about 0.2. The concentration of nitrate in groundwater averages less than 0.1. These are all data from Shale Hills. And the pore water spans this whole range that the stream does, soil pore water. So this really indicates that there is nitrification going on in the ecosystem, right? Because otherwise there's no way for like atmospheric nitrate to generate a nitrate concentration that's like this, right? There has to be nitrification going on, enriching the soil pores, which then enrich the stream in nitrate. So there seems to be a source of net nitrification somewhere, sometime in the system that we should find in order to better understand our nitrous oxide fluxes. Yeah.
Sorry, so are the solar concentrations there for that time period only, or do they always vary that much depending on?
Yeah, I don't know the temporal dynamics of the pore water, but I did put them in fall, winter. I put them in winter, right? Because that's when we're collecting the water. So I actually don't know if the pore water, so like in July and August, we can never sample pore water. So I can't do that exactly. So but the pore water data we have are going to be like from September, October, and November. Is it late? What did I do?
You also said, I'm sorry. You also said in the summer, it's the trees and plants are using a lot of nitrogen, so it's probably not that important. The summer samples, right?
Yeah.
It's probably wouldn't contribute to the storage of nitrogen in the water that much.
That makes sense, unless somehow it's accumulating in dry soils and then gets flushed in the fall. That could be an important process.
Okay.
Ken, did you have another question?
I would comment that the list of nitrogen puzzles is interesting. It'd be nice to stick that on the wall somewhere.
Okay.
And that's kind of what we were talking about going to the all hands was kind of get these puzzles out there. Like, what would you like someone to measure, or what would you like to be able to answer?
So that's what we want. Those kinds of plots, where you got a bunch of different things there, and
but I wouldn't know what the puzzles are.
That's that's all I had. That's my last slide.
So any last questions? We did go kind of late here. Kind of a marathon of nitrogen.
No, I think it's good to.
Yeah, so let's thank Jason and Lily. So the next one we have, I think, is a multiple like this one as well, right? The next seminar. And so I think Tess is kind of in charge of that one. Let's in the future, let's put someone in charge of getting the timing coordinated because we didn't really, because it's a little long, that's all. But this was really good. I think this was a really good companion and everything. And all the slides are going to go up on the box file now? I think they're going to go on our website. And yeah, you put the slides on our website? No, I know, but I think we should put the slides. We used to put the slides up on Angel. So now we're going to put them in box, right? Okay. Thanks, everybody. That was great. Try to put all the top slides.
And that offers you to build this way. That'll move